Hey guys, welcome back to another video under the section of biological databases of computational biology. So we'll just move ahead from where we left in the previous video. Let's get started with this. So this is where we left in the last video. So we just talked about how many types of databases are there, under which categories. So we talked about nucleic acids, we talked about proteins, we talked about structure, genome and others as well. So we talked about gene bank, DDBJ, just a bit of framework. We'll talk in this many more details. All right, so we'll talk about each of them in particular details. So moving on with so talking about sequence database, so which are the two types, the nucleic acid and the protein database. So as I previously mentioned, so there are nucleic acid database sequences such as for DNA and RNA, and protein database sequences for amino acids. So in under nucleic acid we have three such as gene bank, DDBJ and EMBL. Under protein we have many such as TREMBL, PGB and many others. All right, so basically, the nucleic acid sequences databases contain primary nucleotide sequence data, which are repositories. The content of these databases are not curated. All right. So, content of these are not curated or not updated regularly. All right, due to some other reasons. All right. So, moving in with this. Talking about the protein sequence databases, which contain secondary and primary protein sequence data. All right, and not the tertiary ones. So talking about these, some are curated, others are just exact from other databases. So moving on with this, uh, let's talk about the nucleotide sequence repositories. So there are three primary centered exchange information on a daily basis, such as gene bank, ENBL and DDBJ. So all three use the same feature table definition. So you can see the symbols for NCBI, EMBL and DDBJ. Moving with this, let's talk about gene bank. So what it is all about. So it's a US primary nucleotide sequence resource, and it is established in 1988, which was maintained by National Center for Biotechnology Information, situated in Bethesda, MD. And also it contains a release section and a new section as EMBL. All right, and also just you can see the link here. You can just type the link on your browser and just you'll reach the desired page for NCBI, and it has a format. The entries has a format that is different from EMBL. This is a very important point to note. All right, so these has a entry point which is different from EMBL, but it is similar to DDBJ. All right, so gene bank and DDBJ, the entry format is same for both, whereas it is different for EMBL. So talking about EMBL, it's a Europe's primary nucleotide sequence resource, and it is established in 1918 in Heidelberg by EMBL, now maintained by EBI. And its main sources are direct submission from individual researchers, genome projects, and patent applications. And this is the link here for EMBL. So you can just type in this link, and you'll reach to the EMBL page. Also, it has format that differs from GeneBank and DDBJ, whereas the DDBJ and GeneBank has the same format, whereas EMBL differs from it. So, talking about DNA Data Bank of Japan, which is GDBJ, which mainly collects data from Japanese activities but accepts submissions from any researcher in any country. And it began DNA repositories activities in 1986, which was endorsed by the Minister of Education, Science, Sports and Culture. And here again we have provided you the link for DDBJ as well, and it has the same format as GeneBank as I previously said. So moving on with the protein databases after nucleotides. So as I told, I told you about the four databases, four major categories under protein database, which is SwissProt, TREMBL, PAIR and Interpro. So talking about SwissProt and TREMBL, so these are developed by Swiss Institute of Bioinformatics and European Bioinformatics Institution. All right. So SwissProt was developed by Swiss Institute of Bioinformatics, and TREMBL was by European Bioinformatics Institution. Also talking about PIR, which is the Protein Information Resource. All right, so it's a collaboration between National Biomedical Research Foundation and Munich Center for Protein Studies and Japan in the International Protein Information Database. So just theoretical knowledge about PID, what it is, how it was collaborated, how it was founded. So you can just give it a read. Also talking about Interpro, it was developed by EBI, University of Manchester, Sanger Institute. All right, so there are some of the symbols for SwissBrot, PID, and Interpro. So moving on with this, so this is a very important picture that you can see. So I have listed all of the differences that I found between the two. So SwissBrot and TR-EMBL are not the same, so these have a fair amount of differences between each other. So this is the link that I provided for SwissBrot. All right, so it is a curated database. All right, whereas TR-EMBL contains translated sequences from the EMBL databases. All right, so it has a lot of differences. All right, so talking about some of the more differences, it aims to provide a high level of annotation such as function, domain, structure, post-transitional modifications. All right, and it is divided into SwissBrot REL and SwissBrot New, and it is not divided into sections based on species. This is a very important point. It is not divided into section based on species, and we should note the data format. I'll show you. Okay, so talking about this, it contains translated sequences from EMBL databases, and it is divided into SPTR-EMBL with sequences that are candidates for incorporation into SwissBrot. All right, so these contain data that are present in SwissBrot as well. Also talking about REMBL that will not be incorporated into SwissBrot. All right, so this is not compatible with SwissBrot, and this is where this is. All right, so moving on. So moving on about PIR, we'll talk about, this is again the link I provided for PIR. So you can just go through the link and get to the PIR page, and where the aim are similar to SwissBrot, and it grows at higher rate than SwissBrot but has a lower level of annotations per entry. So annotations play a very important role for recognition of particular sequence. All right, so it grows but at a higher level than SwissBrot. That is a huge thing. Also, PID consists of 4 sections. That is very important. So it has fully classified entries, it has verified and classified entries, it has unverified entries, and fourthly it has uncoded or untranslated entries. All right, so moving on with, so talking about Interpro, so this is the link that I provided here for Interpro as well. So it's an integrated database of protein families, domains, functional sites in which identifiable features proteins can be founded applied to known protein structures. I know there's a glitch, you can see this. All right, so no worries about that. So I read it out for you. So which identifiable features proteins can be found applied to known protein sequences. So moving on with this. So talking about protein structures, about primary, secondary, tertiary, quaternary. So you have me. I hope you have read in your. I'm very sure that you have read in your primary classes. So let's talk about primary. It's the order of amino acids. It's basically just the simple order, the sequence of amino acids. Secondary consists of conformations, alpha helix, beta sheets, and lot many complexes. Tertiary is the complete 3-dimensional folding of the polypeptide. Basically, it's the folding of secondary structure which leads to the formation of tertiary structure. And quaternary structure exists if the protein is composed of 2 or more polypeptide chain. So it's like a bit not much differences between each other. See. So talking about structural databases, so it contains information on the 3-dimensional structures of proteins, chiefly proteins, and data is primarily based on X-ray crystallography, NMR, and theoretical models. Examples of this I can see: protein data bank, molecular modeling databases, and links are provided as you can see on the screen. So let's just keep this video till here. So that's it from my video. So thank you for watching this video. I'll be creating many videos under this section, under this subject, under this topic. So thank you for watching. Stay tuned for more.